Hello and welcome back to my channel, The Curious Biotechnologist. If you have not subscribed yet, please hit the subscribe icon below. This is the second video of the series, Blood Decoding the Red Fluid of the Human Body. In this video, we will be looking into various information related to blood, like what is a blood group, what are different types of blood group, how are they identified, and what all kind of information can be derived from a tube of blood by bringing in science and technology together. If you have not seen the first video yet, please check it out after this one. How a blood group is identified? By subjecting a tube of blood to any blood typing equipment that uses a process called agglutination, or in simple words, clumping, which involves mixing of red blood cells or RBCs with various antibody solutions. For example, if the sample of interest has A antigens on the RBCs and the solution contains anti-A antibodies, they will clump together and the blood group will be A. In case if the sample does not react to any of the antibody solution, the blood group will be O. But what exactly is a blood group? Antigens expressed on the RBC determine an individual's blood group. The membrane of each RBC contains millions of antigens. An antigen is any substance that triggers an immune response in the body and brings the immune cells in action. The immune system produces antibodies that are present in the plasma against whichever blood group antigens are not found on the individual's RBCs. For example, individuals with blood group A will have anti-B antibodies in their plasma. Individuals with blood group O will have no antibody, so on and so forth. Blood group antigens can be either sugar or proteins. The antigens of ABO blood group and HH group are sugars, whereas antigens in the RHD blood group and few other blood groups like KID, KEL, Duffy, MNS and Diego are proteins. The more recently discovered blood groups are generally named after first known patient who has produced antibodies against the unknown or newly discovered antigen on the RBCs like KID, Diego and Duffy. A person's DNA determines the type of sugar antigen or carries the information for producing a certain protein antigen on the RBCs. AB blood group is considered as universal receiver because individuals with the AB blood group has both A and B antigens but no anti-A or anti-B antibody in the plasma, whereas O blood group is called universal donor as individuals with O blood group has no antigens but both antibodies, anti-A and anti-B in the plasma. Interestingly, the HH blood group contains H antigen found on mostly all the RBCs and is building block for the production of the antigens within the ABO blood group. If the immune system comes across an antigen that is not found on the body's own cell, or better known as foreign antigens, it will launch an attack against that antigen. But the same immune system will normally not attack antigens that are found on the body's own cell, or better known as self-antigens. What kind of information can be obtained from a tube of blood? A plethora of information can be obtained just by enumerating the different type of blood cells from a tube of blood through blood test called complete blood count, or commonly known as CBC, using a hematology analyzer, a great example of perfect amalgamation of science with technology, which mostly employs electrical impedance and flow cytometry. A CBC result normally provides information like RBC count, hematocrit, which is percentage of RBCs in the total blood volume, and hemoglobin, which is iron containing protein molecule present in RBC, white blood cell count and platelet count. Any deviation from the normal range for these cells direct towards some issues or abnormality associated with the body. A higher RBC count or hematocrit or hemoglobin provides an indication towards heart disease, whereas a low count is mostly linked to anemia. A low WBC count provides an indication towards autoimmune disorder, bone marrow abnormalities, and cancer, whereas a high WBC count is an indication towards infection or inflammation, bone marrow disease, or immune system disorder. A high or low platelet count both indicates towards underlying medical conditions. That being said, merely counting the number of cells in the blood tube is never a standalone diagnostic test for any medical condition. Several additional, more specific tests have to be added onto the test menu for confirming the suspected diagnosis of any disease or abnormality. In terms of more specific test menu, there are several techniques or technologies that are being used currently to identify various medical conditions. For example, blood protein tests are generally performed to detect the presence of abnormal immune system proteins. When detected in higher amount than usual, could be indicative of blood cancer like myeloma. A blood smear is prepared on a glass slide using few drops of blood to study the morphology of different types of blood cells to detect any abnormal cell in the population. Other tests that are very commonly used for cancer diagnosis, called tumor burden or tumor marker test, detects the chemicals or markers that are exclusively or non-exclusively released by tumor cells that can be found in the blood circulation by a PCR or polymerase chain reaction quantitatively, which is amount or concentration of the chemical, and or qualitatively, that is the presence or absence of the marker. That being said, some of the tests are controversial as tumor markers have found to be released by some normal cells in the body with elevated levels even in non-cancerous condition. Another cancer-related test that uses blood is circulating tumor cell test. This test detects the cells that have detached from the primary tumor and swim in the blood potentially to reach any other target organ where they can attach and form secondary tumor by a process called metastasis. Such tests mostly use the principle of immunofluorescence, where an antigen found on the surface or in the transmembrane space of any tumor cell is detected using any antibody specific to that antigen using a fluorescent tag attached to the antibody, which can be detected using a set of filters that can detect the light emitted by the fluorophore using a microscope. So far, only one such test has been approved by the US FDA for monitoring of certain types of cancer. Thank you for watching the video. If you like the video, please hit the thumbs up icon and do not forget to subscribe to my YouTube channel, The Curious Biotechnologist. Stay tuned for more videos. Stay curious and happy learning.